Good morning, everybody. My partner Andres Calle Meneses and me, Federico Buenahora Restrepo, are going to present you our seminar based on the article The Effect of Edelphosine on GRA1 and MIC3 expressions in acute toxoplasmosis.
Plasma is an obligate intracellular parasite with limited aptitude to survive outside the cell during infection. Therefore, involving a new host cell is crucial for the survival and infection progression. The process of adaptation with a new environment has occurred rapidly and requires gene expression changes. Toxoplasmosis can cause mild and asymptomatic infection, as well as a fetal infection that mostly affects the fetus. Is usually transmitted by cats and other felines.
Edelphosine is a synthetic acyl lysophospholipid, or ALP, which is incorporated to the cell membrane but does not approach to the DNA. It has anti-neoplastic effects, so it can be used as a prototype of synthetic lipids against cancer. It also has immunomodulatory properties, which makes that edelphosine can affect the HIV, for example. It also affects the gene expressions. Even though it does not approaches to the DNA, what it does is that it has an effect on the transcription factors. It makes selective apoptosis. Selective apoptosis can be very useful as an antineoplastic effect, and it also is an antiparasitic, which is the thing that can be very useful in this article.
The objective of this study is analyze the effects of edelphosine as a PA-PLC inhibitor levels of GRA and MIC3 mRNA and protein expressions as the markers of dense granules and microsome organelles in acute toxoplasmosis models.
To start with the materials and method used, we're going to explain the animals and the parasites. The animals were some 6 to 8 week old female Baltzine mice that were bred on pathogen-free conditions. They had a 12-hour light, 12-hour darkness cycle, and they had a normal diet. The parasite was the Toxoplasma gondii, and it was maintained in the mice by some intraperitoneal passages. It also was injected to the mice so it can prepare the fresh tachyzoites. RT-PCR. What they first did was to extract the total RNA from the Toxoplasma gondii in the tachyzoites that were cultured in the Piro cells. They then made a chain polymerase reaction to evaluate the GRA1 and the MIC3 mRNA expressions.
In vitro method, first kidney fibroblast bird cells were cultured and prepared in a culture plate. Then they prepared edelphosine and it was applied to the tachyzoids which were divided into subgroups. It was put on one group at the moment of the infection and the other group put 24 hours later. Instant blood method is a laboratory technique used to detect a protein specific in a blood or a tissue sample using electrophoresis. A proteases inhibitor was added to the cultures as a buffer. The extracted proteins were admitted to gel electrophoresis. Then the sample was transferred to a membrane, which was incubated with anti-GRA1, anti-MIC3, and anti-SAG1. They also added IgG. It was visualized with chemiluminescence and the results were quantified.
The results of the effect of edelphosine on the GRA1 expressions on the mRNA, we can see that the lowest concentration of edelphosine had a very significant effect 24 hours after the cell infection. And we can see the more edelphosine we applied, the higher the effect, the higher the inhibitory effect of it. Also in the protein, we can see that the lower concentrations, such as 1 and 5 microliters, were not as significant as the others at the moment of the infection. However, 24 hours after the cell infection, it also had significant effects in this image, we can confirm that the higher the concentration of edelphosine was applied, the lower the expression of GRA1 was shown. We can see that when they applied 0 microliters, the whole protein was expressed, but the higher the concentration, the less you see the protein. I think it's important to clarify that you don't see any change here on stack 1 because this is just a control test.
3 edelphosine significantly decreased the concentration of MIC3 mRNA 24 hours after the cell infection. Treatment with edelphosine at the same time as the infection cells also significantly decreased MIC3 mRNA. 24 hours later, the effect of edelphosine on MIC3 is more efficient. The graphic 4 shows recording the expressions of MIC3 protein both 24 hours after and at the same time treatment by adding edelphosine significantly decreased. In the electrophoresis on the MIC3 protein, we can observe that the higher the level of the edelphosine, the lower the level of the MIC3, which gives us to understand the inhibitory effect to edelphosine on a positive protein such as MIC3.
Now, the discussion. The first author we chose were Lovett and McCoy, and what they said was open quotations. It has been shown that the PIPLC pathway through IP3 and calcium and subsequent calcium-dependent protein kinase signaling cascade has an important role in micronym exocytosis, close quotations. This matches the article because we know that PIPLC is present on both MIC3 and GRA1, and the edelphosine inhibits this PIPLC with calcium. Now, the next author we chose was Oliveira and Lux, and what they said was open quotations. Studies has shown that both metelphosine and edelphosine inhibit the PIPLC lipid metabolism and remodeling the glycosyl phosphatidyllinositol biosynthesis signal transduction in a dose-dependent manner in the disease, close quotations. This also matters because, as we said previously, the edelphosine inhibits the PIPLC, which is present in many proteins, including the ones of interest in this article, MIC3 and GRA1. And now the third and last author we chose was Asus, and what he said was open quotations. The edelphosine, metelphosine, and imosphosine can be effective treatment options in parasitosis through direct action on the parasite membranes. This also matches the article because, as we saw in the results, the higher the doses of edelphosine we give to the patient, the more inhibition it makes of those proteins that can be expressed on the disease.
In conclusion, the importance of this type of articles in medicine or other health science relies on the fact that based on the results and investigation substance such as edelphosine can be very useful to the treatment of disease like toxoplasmosis on other virus, other protozoas, other parasites, because it was shown that common doses of edelphosine had very significant effects by blocking or inhibitor fact the gene expression of the disease, whether it was at the moment of the infection or time later, in this case 24 hours later. The second conclusion, this can also be used as a model of investigation for other disease and other substance, so it's a potential base for medicine to find cures and treatment for various pathologies. The third conclusion is make no laboratory methods implemented in projects to search for answers to scientific medical problems.